So you're used to seeing the furanose where we have, it always looks where the oxygen's at the top and it looks like this. Okay, something like that. There's some type of furanose, and there would be other alcohols and stuff all hanging off. Okay, well, reason why I brought that up is whenever it's a glycosidic bond that's made from furanose, it's called a furanoside, and pyranose becomes a pyranoside. Just some more vocabulary. Yeah, this chapter, unfortunately, has more memorization than probably any of the other chapters. This one, and there's some for nucleic acids and lipids, but they still have enough logic and stuff behind them that it's not completely memorization. Okay, this is important. I do not memorize this table. Okay, I would give you this table, and I could, for example, I could ask you, and the reason why I'm telling you this is later on I'm going to talk about what it looks like whenever we start making these polysaccharides, usually disaccharides. And I would give you a table like this rather than having you memorize all of these, to where you just have to learn to recognize what it is and how to put them together. And we'll see examples of it later on, what I mean by that. But we have a whole host of different possibilities here. This is just starting with glucose. Okay, so you can have a phosphate. You can actually add phosphates anywhere, and that's really important biologically speaking, which unfortunately we don't have a lot of time. We probably won't. I was looking at trying to gauge where we're going to be at the end of the semester. I don't know that we're going to be able to get through chapter 12, which is biosignaling the cascade pathways, but these phosphorylated sugars are very important for it. In particular, there's one called inositol, which is also a sugar, and the position of the phosphate on there, whether it's 1, 2, 3, 4, 5, 6, or you can even have 2 or 3 or 4 or 5, even all 6, which they abbreviate as IP6, can be phosphorylated, and it really matters because it has a different functionality in the cascade. So that's why it even matters what position. This is at position 6, but you can have it at any of the other positions as well. Okay, and then of course you've heard about glucosamine. I even take glucosamine as a supplement. What's it good for? Does anyone know? What do they say to take glucosamine as a supplement for? You guys are too young to have to worry about this, usually. Like your grandparents will probably take it, or maybe your mother. Glucosamine is for your joints. Glucosamine and chondroitin. And so, but yes, and you can just see that's literally where they've replaced one of the alcohols with an amino group. Now this one, and we're going to hopefully we'll have enough time today to cover it later on in greater detail, but this one is very important biologically, and so is this one. They abbreviate them as NAM and NAG, NAM and NAG. But what you can see here is in this one, if you think back to organic chemistry, it's N-acetyl beta-D-glucosamine, because it was glucosamine. Whoops. Oh, on mine, you can't tell it, but I'm covering up the others. Of course, it just highlights it. But if it didn't have the acetyl group there, it'd be glucosamine. And then it does have the acetyl group added onto it, and you have to give it that locant, the N. Then, just one slight difference, by adding this larger group here in place of another alcohol, it becomes N-acetylmuramic acid. Okay, and so, which those are important later on. They're used in for antigen response, especially with respect to bacterial cell walls and cell membranes. But you can go on, like I said, you can have lots of different possible glycosidic bonds and different types. And I would expect you to know this one can still do hydrogen bonding. It could also act as a nucleophile, the nitrogen could, whereas this one right here is huge and negatively charged, so it's going to make the sugar larger and more negative, things like that. Okay. I forgot to follow along. Oh, stupid computer. There we go. All right, then we have the phosphoric esters. So these are really important for the breakdown. We're going to see this glucose 6-phosphate. You're going to hear about it a lot next semester, which they like to use different types of abbreviations. The sugars themselves have abbreviations as well. And so glucose would be like, Why is it not wanting to work? GLC6P, or sometimes a lot of times I will put a P in a circle so that way you can tell it's a phosphate versus just a phosphorus. But those are the kind of things that they talk about. But we're going to find out that this is the first step in glycolysis. Okay, and so we're going to find that these phosphoric esters are really important. Another really important phosphoric ester that we'll talk about in greater detail in chapter 8 is this one. And by chapter 8, I expect you to know how to draw this. Oops, I don't have my pen, and so, well, it's supposed to be in the joint. Oxygen, it goes on up. This one goes on. Why is it not wanting to work? This one goes on down. Okay. So this goes on down. That other one goes on up. So what is that one? That's an important phosphoester bond. What is that? What is that sugar? What is it used for, and what biomolecule? That one's RNA. Mm-hmm. So that's the sugar backbone. Of course, this would just be the base, so it's going to be an A, C, U, or G. Or you're going to find out that there are other bases as well. That means because it goes down to the next base. So because it's part of a long chain, just like you can't tell, but there's the oxygen on the top also would have a squiggly to show. Also, it was just a single mononucleotide. Okay, and we know it's an RNA because it has the alcohol group here. If it was a hydrogen group there, then it would be DNA. And I know in biochemistry that we don't do this in organic, but a lot of times biochemistry, these are not methyl groups. Those are hydrogens. You
So
just get sloppy.
connect on any of those three locations? The oxygen down here?
So this is what makes RNA different than DNA. If DNA, this is a hydrogen. It's got to be, remember, it's 5' to 3'. This is the 5' carbon. This is the 3' carbon. Because it goes 1, 2, 3, 4, 5. So that's where the directionality comes from.
You said that base, where you wrote base.
That's like where the adenine, guanine, cytosine will be. And I don't have you, by the way, when you get to that point, I don't have you memorize. I want you to know how to do the backbone, but I don't have you memorize all of the structures for all the bases. Okay. I didn't even do that. That's not good. Get out of the way. So we can also have amino sugars, which are really important. That's just where, once again, one of the alcohols has been replaced by an amino group. And then this one is, the one on the left is sometimes I call it NAG, for N-acetyl beta-D-glucosamine, and then one's NAM, which is really funny because one of the labs that I worked in, that I got my PhD in, especially at Duke Medical School, but then we moved on to the University of Michigan Medical School. But one of the projects that was going on in our lab had to do with looking at antibiotics, making broad-based antibiotics, and they base it off of the sugars that get added on the cell wall of the cell membrane, and these are in the big chains that get added, and so they just call them like NAM-NAGs all the time. So they'll say that NAM-NAG, NAM-NAG, all that kind of thing. And we just always, those of us who didn't work with the sugars always laughed. NAM and NAG. Okay, this is not the important, but this is a very important part, and this is the kind of thing that I was talking about that I would expect you to be able to draw later on. I would tell you give you the structure, what the glucose looks like, but I would expect you to know how to do the linkages here. Okay, so this is a disaccharide. This is maltose, which is a sugar. And this is 2 beta-D glucoses that are hooked together. It's always a condensation reaction, and to break it apart, it's a hydrolysis reaction. So condensation means you're going to give up a water, so you're linking you just have one linking oxygen, of course. Hydrolysis means you have to add a water to break them apart. But this is really important because they actually tell you a lot here. Okay, so First of all, I want to point out that just like organic chemistry, the YL, whenever we had YL like phenyl and benzyl and what were some of the other ones? Now I'm having a complete break. Acetyl and things like that. What did the YL mean? What did it indicate? Heck, it's hanging off. And so that literally means that this group right here is hanging off of whatever the parent is. And they give it a directionality just like this. Okay. And this literally tells you what carbons are connected. So that's why the sugars make it very simple. It's the carbon number 1 is connected to carbon number 4. So it's 1 to 4. This is the first one. This would be the second one. Of course, we've already talked about what alpha versus, beta would be. And so we know this one's the alpha because you look at the scorpion's tail, and if the first alcohol that it was starting off on, or on the anomeric carbon, I should say, is going down. So on the opposite side of the scorpion tail, then it's alpha. So this is the alpha glucopyranosol. And then it's a 1,4 linkage to glucopyranose. Okay, and so we know now this is carbon 1, 2, 3, 4, and so it's a connection. And so we see this is the cartoony way of showing what an alpha connection looks like. Okay, so now we're going to practice. And if you've already printed out the slides, please do not cheat because I want you to be able to practice looking at these. So now we're going to talk about some of the other disaccharides. This is sucrose. Sucrose is table sugar. Yeah.
Just on the previous slide,
Mm-hmm.
for the second part, the parent sugar, well, it said D. So I'm just a little confused as to why the second one was a D and not an F.
It's based off of the original Fisher projection.
Okay, off the original one.
Mm-hmm. And
So if they're opposite, it's D.
No, opposite's alpha and is alpha.
Okay.
D is back whenever it was not a cyclical part.
Left or the right?
It told you the left or the right. Mm-hmm. Yep. Which almost all, I'm sure that there are some exceptions, but almost all of the naturally occurring sugars our body utilizes, I do believe is a D. I know it is for glucose and fructose. Okay, this is sucrose, which is table sugar. It comes from sugar cane or sugar beet if you're up north. Okay, and so it's literally a D-glucose with a D-fructose joined. And so I just want you to practice now saying, first of all, is it an alpha, beta, if it's an alpha or a beta linkage, and secondly, what the number is. Okay. So I'll give you a moment to look at it before we look at the... And the only difference between the one on the left and the one on the right, the one on the left is like the traditional way of drawing the pyranose and the furanose. The one on the right is that chair formation if you think back to organic. And so it's the one that more accurately reflects the three-dimensionality of it. Sorry about that. Okay, so if we look at this one right here, the glucose, first of all, was it originally an alpha or a beta? It's alpha. Okay, so that's half of it right there. So we know that this is, I don't remember where it's an alpha. And now we have to put down a lot of times we put it in parentheses. Sometimes they don't, but I'm going to, so that way try to be as obvious and consistent as possible. So what carbon is it in the glucose that is attached to the fructose now? What carbon number? 1, 2, 3, 4, 5, or 6? What carbon is it? It's carbon 1. Mm-hmm. Then the way that on the previous slide that showed you a little arrow, you're going to see in a moment, some of them are like in organic chemistry where sometimes they use a comma, but I'm just trying to be consistent. Now on the fructose down below, what carbon number is it attached to? And I don't know why there's that V there. Just ignore the V. The V has absolutely nothing to do with it's part of the figure that came through. And so this one, and I would say that fructose is probably one of the most difficult. And so it's one of the reasons I also picked this. And I'm going to actually show over here, show it on this one because it's a little easier to see, a little less things. So this is either carbon number 1, so 1, 2, 3, 4, 5, 6, or this one's carbon 1, 2, 3, 4, 5, 6. Okay, and it's carbon number 2. So that's an alpha 1,2 linkage. And so there's the answer, and that's why at that time they used commas, arrows. Right, so here's another example. This is lactose, which many of us are lactose intolerant or have some degree of lactose intolerance, and it can change as you get older. There's also differences between various ethnicities and cultural groups and things like that as well. Okay, so this one has a galactose and a glucose. And once again, I would give you the structures of the original galactose and glucose, and just either I'd give you this and ask you what kind of linkage it was, or I'd give you the name, like galactosol something something glucose, and ask you just to draw it. And once again, the only difference between these two is one of them's the chair conformation, and one of them's the traditional pyranosol conformation. Chances are you'd probably draw the one that's on the left because I copied this from a table. I'm not going to draw these out myself, and I'm not going to ask you to draw it as a chair. But if the table I gave you had it in chair, you can draw the chair. It doesn't. Not too worried about that. Okay, so we start with the galactose. Okay, and on the galactose, first of all, was it originally an alpha or a beta linkage? Say it. What's conviction?
Beta.
It's beta. Okay, and what carbon number is it? So the way you do it is look for the oxygen that did the initial attacking to make the cyclical pyranosal group or furanosyl group, and see what carbon that it attacked, and then see. That's going to be the lower end, like 1, 2, or 3. It depends on how big the sugar is. So what carbon is it in the galactose that's in the link? Is it carbon 1, 2, 3, 4, 5, or 6? It's just carbon 1. Once again, I'm going to do it with the arrows.
Which region are we looking at?
Pardon?
In order to determine that it's beta, which hydroxyl?
So to determine whether it's beta, so here's the scorpion's tail. This is the anomeric, is it coming up on there? Yeah, this is the anomeric carbon. So that was the alcohol hanging off the anomeric carbon. See, otherwise, if it was alpha, it would be hanging down. Okay, now in the glucose, what carbon is it attached to? Is it going to be 1, 2, 3, 4, 5, or 6? So this one's going to be 4, because you have to look on the original, the way that they do the numbering is based off of the original oxygen that did the attacking, and it attacks the lower, I can't think of any exceptions, it attacks a lower number carbon, so it usually is going to be carbon 1 or 2. I'm sure that there's probably one weird one out there somewhere, but usually chances are this is either always going to be carbon 1 or 2, the anomeric carbon. Okay, so that's carbon 1, 2, 3, 4. So this is a beta 1,4 linkage. Or a beta 1,4 glycosidic bond. Here's the answer. It's prettier than mine. Oh, I think we already did this one. I think I skipped ahead. I already did this one, but we can still practice again. Okay, so this is maltose, which are 2 glucose units. And first of all, if we look at this glucose right here, was the original sugar an alpha or a beta? Pardon? It's alpha. Okay, and what carbon was it that did the attacking initially to make the bond? I shouldn't say the carbon. It wasn't the carbon that did tagging. It was the oxygen. But yes, it's carbon 1, and it's attached to what carbon? That ether attachment now is on what carbon of the next glucose. And so we start here. Remember, there's the one that did the attacking. So that's carbon 1, 2, 3, 4. So it's a 4. Because this one right here is 5, and that the scorpion tail and glucose is 6. So, all right. Just like, and don't memorize this, but I just want to let you know, in case some of you may have been using enzymes in your paper that have sugars in them or whatever, there is a code, just like we have a 1-letter and 3-letter code and all that for amino acids. There's also a code for sugars, and they also like to use little cartoony pictures a lot of times to make them, and so that's why that just shows you, so for in this example, this star would be xylose, and its 3-letter code would be XYL, sorry. I've already mentioned glucose, and that is one that you'll see a lot next semester, glucose, maybe galactose.
You said this is something we don't have to memorize?
No. I would, next semester, you see GLC long enough times that you're going to know it's glucose. That, and it's not an amino acid. Oh, and fructose is the other one that you see a lot next semester. Okay, but just to let you know. So then there are different ways to make the polysaccharides. Now we're talking about not just 1 or 2 coming in. We're talking about they can literally be hundreds, if not thousands, long. So 2 of the broad categories are you can have ones that are Homo Polysaccharides, which is the kind right here, versus hetero. Homo means it has all the same. So maltose is a very simple, but it was one that was a Homo disaccharide. And then you can also have it either unbranched or branched. Okay, so on both of them. So it's just a way. I mean, I think it's pretty simple. Some of my favorite enzyme names for this come from the metabolic pathway. And then I just wanted to give some examples here. So this is cellulose. This is the kind of course, cellulose is what gives the structural part to plants. This is the cartoony way that's often drawn. Here, let me just blow it up. And it's fine with me. If I would give you the sugars and I asked you to draw me a beta-1,4 linkage, it's fine if you draw it cartoony like that rather than trying to draw it at a weird angle. Either way is fine. That's a lot of the way that they do the beta linkages. It's little curly Q's versus the ones that look like U's for being an alpha. Since an alpha started off down and a beta started off at going up, it's perfectly fine. So this is a beta-1,4, and it just repeats over and over and over again.
So how do you know if what's connected to is alpha or beta?
It only...
Do you know, like it wouldn't be beta to beta, like it...
No, because that alpha and the beta only matters on that carbon number 1 or 2.
Okay, so if we drew the cartoony like up and up, you see like...
Then up and up would be a beta, I'm sure. Well, it depends on where you put the scorpion's tail. If you put the scorpion's tail here and you had it looking like that, then that could be beta, I'm sure. Yeah, because the original oxygen would be going up.
Yeah,
that's
so
why it
the
dictates it.
one that mattered.
Mm-hmm. So that's why then you have the same linkage over here. It's not always the exact same sugar. In this instance it is, but that's just to show you. Oh, whoops. I meant to resize this so that way you can see it. The reason why this is so strong, even though they're just really, really, really long chains of the same thing over and over and over again, you can imagine that every single one of those alcohols can do hydrogen bonding. So each fiber, so to speak, I guess they are fibers, each chain can hydrogen bond to the next chain over, can hydrogen bond to the next chain over. So because of that, that's what makes it so strong. You can see, yeah, we're talking like 2,800 of them in a row. So there are lots and lots of hydrogen bonding, lots of covalent bonds within the actual fiber itself. All right, here's some other examples. So this is starch. Starch would be alpha D glucose. Once again, for the amylose, I could ask you to draw an example of an alpha 1,4, two glucoses in a row for alpha 1,4. And I'd show you what a glucose looks like. You would have to draw an alpha 1,4 linkage between the two. So that is not uncommon to see something like that on, an exam or a quiz, just because of the fact there's only so many things that you can ask. And it matters depending upon, so the only difference between, I thought it was on the other one. The only difference here between amylose and amylopectin is the fact that amylose is just a 1,4 and it's unbranched. Amylopectin has branches. And so not only is it 1,4, but then it also branches off at position 6 and has 1,6s. So, and this is going to look really bad. It kind of looks like this. Let's see what alpha. Whoops. They're supposed to be touching. And then you'll have another one popping up off here. There are much better pictures coming up. All right. So the enzymes, I don't think anyone this semester is using these. I can't remember. The enzymes that catalyze the 1,4 linkage, so that would break down the chain here and here, are called amylases. So where's one example, one part of your body that has a high concentration of amylase? This is from A&P and biology. What? It's in your saliva. So, oh my goodness, sorry about that. Oh my goodness. Then, but see, amylases can't do that 1,6 linkage. They are very specific. It breaks down 1,4, 1,4, 1,4, but there's another enzyme that has to break down that 1,6 linkage. It has one of my favorite names for an enzyme. Does anyone know what enzyme that one is? Here, let me see if I can go back real quick. I'm going to pause this. And the reason I say that is because it's called the debranching enzyme. And that's what all it does is it takes off, and we'll talk more about it next semester when we go over metabolic pathways. But since amylase can do 1,4, 1,4, but it can't get it gets so far and it gets stuck because it can't handle the branch, debranching enzyme comes, it breaks that one, and rather than just breaking it, what it does is it breaks it off 2 or 3 ahead of time, and it sticks it at the end of the next chain down, so that way amylase can come down and go through and break it up even more.
So it basically makes it a 1,4?
Yes. See here, I'll show you there in the picture. So that's amylose versus amylopectin. And so what debranching enzyme? So amylase, has no problem with this one. Amylase, when it does this first branch, it gets I want to say 3 up. It's either 2 or 3 away from the branch, and it gets stuck. The enzyme doesn't fit. It'd be like hitting its head against the wall. So the debranching enzyme then comes in, it just takes it, and I think it's cool because it just breaks this one bond here, and it sticks it at the bottom on this one. On the one just below it, the branch is below it, and so that way amylase can then go on and chomp till it gets into the branch. And debranching comes in. And by the way, the opposite of debranching enzyme, the one that whenever you have to build these structural sugars, it's called the branching enzyme. There's branching and debranching. They also have official formal names, but everyone calls it branching and debranching. Okay, and so then we have chitin. This is just another example. This one, though, is not with just glucose. So even if I had hidden this, which next time maybe I will, I would expect you to be able to figure out that was a beta 1,4 linkage. And if I gave you the answer key, like the key of all the sugars, I would expect you to be able to figure out what sugar that one was. And you say, oh, that one is N-acetyl D-glucosamine. So the bacterial cell walls, I always try, usually, especially whenever we have more time throughout the semester, I always try to bring in even more biomedical relevance. So the bacterial cell walls are made up of these repeating NAMNAG units. You just see the same thing over and over and over again. If you think back to Gen Chem and organic, this is just showing you that it's a polymer. The N means it goes on and on and on. It repeats over and over again. So if we were going to, first of all, is this an alpha or a beta linkage? Is it alpha or beta?
Beta.
It's beta. And what carbon number is this one that we started off with? 1, and the carbon on this side is 4, so it's a beta 1,4 linkage. Okay. See, beta 1,4. See, I have problems saying that word, muramic acid portion of it is this portion down here, which has a carboxylic acid and a carboxylate, just like your amino acids do. You know, it has a carboxylate end. So it can actually be used, and it's used by the bacteria, to add amino acids to it. So the reason why that these are so important in the bacterial cell walls is they are able to take these and further modify them. So for example, we can take that oxygen and now start adding on larger things. Here, let me go back and I'll show you, do a better job. We'll see how well that works. Okay, so that's why we formed a new peptide bond. And so now there's a peptide bond between an actual group of amino acids and sugars. So this is one of the ways that sugars can be added and modified onto proteins. But what happens here, and these, I mean, it's cool, but it's also really bad for us, but good for the bacteria, but bad for us, is that they have these four amino acids in a peptide bond that go on down. One of them is a lysine. If you remember, lysine's got a whole bunch of carbon, hydrogens, and then that one amino group there. Well, that one amino group there, the epsilon amino group, is then used to make a new peptide bond going the other direction with 5 glycines, and those 5 glycines can actually be then formed to another peptide bond to another group of 4 on another side chain. So it makes the bacterial cell wall extremely, extremely tough. That's the reason why we have, not only do you have sugars, but you've got amino acids forming, they're not really proteins, but they form these amino acid bonds that help hold them in place, okay? So this is what it looks like, this little cartoony way. So here we have our NAMNAGs. Then hanging off of one of the sugars, you've actually got 4 amino acids. Hanging off that lysine, you've got 5 glycines that can then now form a new peptide bond to another group of 4 that's hanging off of another whole line, and so it just adds increased strength. And that's why it's called the peptidoglycan cell wall, because that literally means peptides with sugars. In my old lab, one of the things that they worked on actually looked at stuff like this. This one's not quite right. It doesn't. I don't want you to think that the fact that you've got the O antigen, you're going to go into and have toxic shock. But I didn't have a good picture of what I was looking for, and so I'm just showing you the blood typing. But bacterial cell walls have something very similar to this in the sense that they've got these really long peptidoglycan polysaccharides hanging off of them. They're called lipopolysaccharides because there's a lipid portion, then there's a saccharide portion, and some of them will even have modified saccharides after that. What happens is this very first sugar is called a Kto sugar, and there's an enzyme, if this wasn't the O antigen, if this was the one for the cell wall for bacteria, there is an enzyme that's very specific for that, called LPXC. I wouldn't have asked you to know that. But one of the experiments that was ongoing in my lab was to look at the mechanism on how that enzyme works, because they were they were in the process of designing inhibitors for it. Because if you can inhibit the ability for the bacteria to put the sugar onto its what would become the cell wall, that bacteria dies. And so in particular, they were looking at this with staph, Staphylococcus. Okay, and also Pseudomonas aeruginosa, which does anyone know why is Pseudomonas so bad? I mean, we have Pseudomonas around us all the time, but there's one group of people in particular that's highly, Pseudomonas is highly lethal for them. It's people with cystic fibrosis. They usually die from problems due to Pseudomonas Aeruginosa. So we actually had money from Cystic Fibrosis Foundation and some other foundations to help look at that mechanism to try to develop inhibitors for it. Because once again, it's called the concomitant step. That's a step that dictated that the metabolic pathway is going to go forward. It was made it committed. That's why it's called concomitant. Okay, but it's actually the sugars that's hanging off of the cell walls of bacteria that causes toxic shock and causes the bad negative effects many times. All right. There are naturally occurring polysaccharides that also have sugars modified onto them. Just some of them to point out. We have heparin. We have the chondroitin, which I talked about before, which helps with along with glucosamine. It's to help with the connective tissue in your joints. Okay, and so this just shows you some of the pictures. Once again, which I didn't hide it here, but I would expect you to be able to say, okay, that's an alpha linkage, that's carbon number 1, 4. Here's 1. This one's different. This is a beta 1,3. I mean, it's already on there, but glycoproteins just means the fact that you have proteins that have sugars that are added onto them. And I always want to, you should have gone over this in anatomy, physiology, probably in biology as well. But this always got lost whenever I go over it in forensic science at, I don't know, the beauty, or if you're fatalistic, or I don't know, how precious life is, or what have you. But this is what dictates the difference between blood groups, okay? Whether when we hear about the ABO system, there's A, the B, the ABO, the A, the AB, the B, or the O, that you have a possibility. And then we have the plus minus, which is the rhesus factor, the anti-D. But the only difference between them are the carbohydrates that are actually attached to the proteins, and that's what's the antigenic determinant for it. And I always want to point out, because we're about out of time, is how similar these are. I absolutely hate this figure right here, okay, because it took me, this was in the old book, it took me forever to try to figure out what they were trying to say. Like, what is this? But this is them showing an alpha 1,2 linkage, and alpha 1,2 from Fructose up there, and so on and so forth. And remember, there's the reducing end, which has the free alcohol, and the non-reducing end. I'm sorry, the reducing end is the one that had the anomeric carbon, and the non-reducing end would not have the anomeric carbon. So I like this one better. But in A, what it is you have an NAG. In B, it's just a galactose. It's just, does it have that N-acetyl group? A, B, they're both present, and in O, neither one is there. So I like this one because it just shows you how similar it is, and it's literally a single acetyl group between A and B. And so if that acetyl group is not there, then bad things can. I'm sorry, it's in N-acetylgalactosamine versus an N-acetylglucosamine in a galactose, right? So that's the only difference. Is if we look here, the only difference between A and B. O is missing that sugar completely, but A and B, A has an N-acetylgalactosamine, B is just a galactose. So it's missing the amino group and the acetyl group, and that's the only difference between the two. And that's what can literally cause the difference between life and death. Okay, and then we actually don't have time to go over low carbohydrate diets here. We go over a lot next semester, a lot, because we go over the metabolic pathways that are involved between low carbohydrate diets, and also how that affects fat storage, fat synthesis, fat breakdown, all that kind of stuff. We go over that in great detail next semester. So normally I just give a little prelude to that, but for brevity's sake, I won't this semester.